Good morning, children. Very glad to meet you. Today's class, children, you are all going to learn an important question. Polygenic inheritance in wheat. In the previous class, we started intergenic interaction in which we discuss about how the Mendelian diabetic cross ratio is modified due to the epistatic gene. We discussed dominant epistasis in which the fruit color in summer squash was discussed, and also we gone through extra chromosomal inheritance. It is also called as cytoplasmic inheritance. Previous year, that is 2019-20 public examination, in which this question cytoplasmic inheritance was asked. Okay, so we continue the chapter 2, classical genetics, in which polygenetic inheritance. Poly, numerous, genes, inheritance, transmission of characters. Mendel recognized that one character is controlled by a gene which contain a pair of alternative form or alleles. Polygenic inheritance means more than one gene that combine to affect a single trait. More than one gene or many genes which contribute together in determining a single character. So several genes which combine together affect a single trait is called polygenic inheritance. In this polygenic inheritance, we are going to look after about kernel color of the wheat. Kernel color of the wheat. Polygenic inheritance. A group of genes that together determine a characteristic feature of an organism is called polygenic inheritance. It gives explanations about the inheritance of continuous character which are compatible with Mendel's law. The first experiment on polygenic inheritance was demonstrated by a Swedish geneticists Nilsson et al. 1909. A Swedish geneticists H. Nilsson Helen in wheat kernel they studied and demonstrated the polygenic inheritance. The kernel color is controlled by two genes, each gene with two alleles. One gene with alleles or one gene with the red color of the kernel was dominant to the white. So there are two phenotypes in the kernel color of the wheat was studied. Each color is controlled by genes with two alleles. He crossed two pure breeding varieties of wheat. They are dark red kernels and a white. He denoted the dark red genotypes as capital R. Okay, so each character, that is the red color of the wheat kernel, is controlled by two genes, that is capital R1, capital R2. Each gene have two alleles. So the genotype of the dark red parent is capital R1, capital R1, capital R2, capital R2. Whereas the pure breeding white is recessive, its genotype is small r1, small r1, small r2, small r2. Both were pure breedings. In the F1 generation, you can see the wheat kernel color is controlled by two genes, capital R1, capital R2. Capital R1 gene has two alleles, dominant red and a recessive form of white. Capital R2 gene also have a dominant form of red and recessive form. The dominant form of the red gene for the red color of the kernel is capital R1, capital R2. It has two alleles, means capital R1, capital R1, capital R2, capital R2. Recessive form is small r1, small r2. Its alleles are small r1, small r1, small r2, small r2. You may understand now by seeing this picture. The two pure breeding parents of dark red wheat variety is crossed with the dark red kernel variety of wheat is crossed with the white color kernel variety of wheat. F1 generation was raised. They shows medium red. The phenotype of the F1 generation is medium red. Its genotype is capital R1, small r1, capital R2, small r2. F1 wheat plants which produce four types of gametes. Like diabetic grass, it produces four types of gametes, namely capital R1, capital R2, capital R1, small r2, small r1, capital R2, small r1, small r2. The color intensity in the wheat kernel is determined by capital R gene in the F2 generation. So the color of the wheat, which is dark red, is determined by the number of capital R gene. How many numbers? If all of them are dominant allele, capital R1, capital R2. So four alleles, capital R1, capital R1, capital R2, capital R2 means it is dark red. So number of dominant gene R is present in the F2 generation determines the color. For R gene, the color of the wheat kernel is dark red. You can see the checkerboard in the slide. In the checkerboard, if you observe, capital R gene is 4 is present, definitely it is dark red. If 3 capital R gene is present, it is medium dark red. The color is medium dark red. If 2 capital R gene is present, it is medium red. 1 R gene is there, the color of the color will be dark red. So 4 capital R gene, it's dark red. 3 capital R gene, it is medium dark red. 2 capital R gene, it is medium red. Only 1 capital R gene in the genotype, it is light red in color. Due to the absence of capital R gene, which is dominant, so the absence of the capital R gene, the recessive gene which produces the color called white. The capital R gene is an additive. It is act as additive manner produce the red kernel color. The number of each phenotype is plotted against the intensity of the color. You can see the intensity of the color in which this represents the distribution of the phenotype. Now you can observe, children, where a graphical representation of the intensity of kernel color can be given. The number of each phenotype is plotted against the intensity of red color kernel, which produce a bell-shaped structure, a bell-shaped curve. This represents the distribution of the phenotype. From the dark red to the white color of the kernel is formed due to the presence of the number of genes. Okay, the number of genes that's seen in the capital R, that's seen in the genotype. Polygenic inheritance also studied in human being. Height and skin color of the human being are controlled by 3 pairs of genes. So what is called polygenic inheritance? Several genes which combine together affect a single trait in an organism is called polygenic inheritance. Finally, in recent where the loci that studied by Nielsen and Hill were not linked and the genes are asserted independently. Remember, the genes which control the color of the color is not arranged in the same loci. So they can be easily get separated from one another due to the independent assortment law. Very recently, scientists discovered the third gene which also affects the kernel color of the wheat. The 3 independent pairs of alleles were involved in wheat color. There are 3 independent pairs of alleles which are involved in color of the wheat kernel in plant. Nielsen and Hale found that the ratio of the F2 generation has 63:1. 63 of them are red and 1 is white. The F1 generation in which it appeared as follows. You can see the slide. The genetic control of the color in wheat kernel, F2 generation, dark red 1, medium dark red it is 4, medium red it is 6, light red it is 4, and white is 1. Okay, the shades of various red color is 15 red and 1 white. 15 red and 1 white. Right? You can see the polygenetic inheritance in wheat kernel. Polygenic inheritance in wheat kernel. The 3 pairs of independent alleles, capital A, capital A, capital B, capital B, capital C, and white kernel is small a, small a, small b, small b, small c, small c. F1 generation is intermediate red itself. There are 63 red and its many shades along with 1 white. From the above results, Nielsen et al. Showed that blending inheritance was not taking place in the kernel of the color. Blending inheritance in the sense the genes or alleles which control the particular trait, even they brought together, they did not mix. The F2 generation plants have kernels with a wide range of color variation in red. It is mainly due to The fact that is genes are segregating and recombining together was took place. Another evidence that absence of blending inheritance is that parental genotypes dark red and white appears in the F2 generation. Due to the absence of blending inheritance, only due to there is no blending of genes, only the phenotype. Genes are brought together, that's all. The phenotype only got changed. The cumulative effect of several pairs of genes during interaction gives rise many shades of the red. That is dark red, medium red, light red, and due to the absence of the dominant gene capital R where it produces only white. So he hypothesized that the two loci must contribute additively to the kernel color of the wheat as white. The contribution of each red allele to the kernel of the color of wheat is additive. The contribution of each red allele to the kernel color is in wheat is additive. This is one important question in 2 marks and 1 word questions where they can ask the phenotypic ratio of F2 generation, various shades of the red color children.